Okay, let's proceed. So we have, let me take, it's motivating to take it. So we have looked at the principal component analysis. And I would like to mention another dimensionality reduction technique called T-distributed stochastic neighbor embedding, TSNE, or TSNE, TSNE, which is another dimensionality reduction technique, but it's a non-linear dimensionality reduction technique. And I'm mentioning it here because it's used more and more for kind of high throughput omics data. It's used a lot for single cell sequencing data. We use it in my group for spatial proteomics data, like this hyperlopic dataset, we use it. So it's a very interesting technique. But it does come with its risks, and as opposed to PCA, it has a couple of parameters to tune. These are the number of steps, the number of iterations that you run the algorithm, and then the perplexity, which is another parameter that controls kind of the if you want the stickiness of points, points that are close to each other, very similar, are more sticky than points that are not as similar. And as you can see, depending on the number of iterations that we have, what's the value at the top? If we have 10 iteration, 1,000 iteration, or 10,000 iteration, and whether we use a perplexity of 5, 30, 50, and 100, the values of these parameters will influence what kind of grouping we have. So we will need to be careful to this. And here is, for example, a comparison of this spatial proteomics data set using principal component analysis and TC. Now, the fact that this dimensional reduction technique is non-linear has a full consequence that we need to be very careful of how we interrogate these results. So points that are very similar are close to each other here, and we can do some reasoning at a very local level. But as soon as the distances are far away, we cannot start having the similar reasoning for points that are far away. It is something we are not able to do with TSNE. It's a probabilistic technique. It's a little bit like if you have a high you do a statistical test, hypothesis testing, and you have a high p-value, you can't say anything whether your p-value is 0.8 or 0.9. There is nothing you can say about this. Similar reasoning. But it is a very interesting technique, which is why I mention it. And you can run this, you can use TSNE in R, and the package is called RTSNE, and the function to use it is also called RTSNE. So you can run this piece of code to generate the two plots with PCA on one hand and TSNE on the other one to compare them. But before we carry on, it's important to realize that these algorithms are all very powerful, but inherent risk when we're with high dimensional data is what is well known as the curse of dimensionality. When the number of dimensions increase, the data becomes more and more sparse. And then when we have very sparse data, the measures of distance between data points stops to make sense as we have in low dimensions. Okay? And more of these points become outliers. And here's one illustration of this. So this code here does the following thing for a certain number of dimensions. Oh, here, dimensions from 1 to 200. And it calculates and here the number of points in these the dimensions. And it so in 2 dimensions we have a square. So we have 2 dimensions, quite easy. We have a square, and so it looks at the, what is it? I think 1%. So the outliers in this square 3-dimensional cube or hypercube. And so it looks like how many points do we find in the kind of outer shell. And then it counts what the proportion is of these outliers. And this is what we see. We see that when we increase the number of dimensions, so, the proportion of outliers increases. And so when we have very high dimensional data, the great majority of our points become outliers based on here. Another illustration of this curse of dimensionality comes from this paper here, where they do the following simulation. They simulate 100 observations coming from 2 classes. So we have a red class and a blue class. 50 observations from red, 50 observations from blue. And they simulate this data in 2 dimensions, 40 dimensions, 200 dimensions, and 1,000 dimensions. And then they do a principal component analysis, and they look at these plots along the first 2 dimensions. And what they see is that despite the fact that the way these data were generated based on, belonging to 2 classes, we lose a lot of resolution for these data points. So if these were proteins, as the number of proteins increase, or the number of noisy proteins increases, we tend to lose signal.
So you need more observations.
Do you need more observations? I don't think so. I don't think having more observations would...
like if you had 500 proteins in 500 patients or samples versus 500 proteins in 50,000 patients.
Yeah, okay. You will... Well, you will not be...
better proteins quantified in the signal verification.
I think this means that more data is not always better. No, we will never be able to quantify 200,000 proteins, but we need to be careful with more data. More data does not necessarily mean more resolution. If this additional data is noisy or doesn't have strong signal, we will not improve our results. Yes?
How do you define noisy?
How do you define noise?
Yeah. We're talking about low signal, low intensity?
So if in a 2-class problem, it wouldn't, they have random, the signal or the features the distribution of this signal is random along your range. So we have a cluster that is defined here. and then maybe so if I have two proteins, P1 and P2, and sample 1 has a high expression in P1 and P2 it appears here. Sample 2 has a low expression in P1 and P2 it appears here. So, these are two proteins with single points. Here, principal component 1, principal component 2. So on average, the distribution along first principal component is low, PC1, PC2 for one class, and high for PC1 and PC2 class. So here we have good resolution. But if the signal, let's say we have another data set where the signal is average for protein 1 and average for protein 2, we will have points that all segregate in the middle of a point, and then it will become difficult to separate them. So this would be kind of noisy data, because we don't, based on these proteins, we are not able to define two well-defined classes. As opposed to here, where we tend to have high signal for protein 1 and PC1 for one group, and low signal for the other group. So we have good separation or bad separation here. So the bigger our data sets are, the more careful we will be. It doesn't mean that more data will mean better resolution. And there is a trend towards bigger data sets and using these more sophisticated algorithms like TSNE, but they come with certain drawbacks sometimes. So now I would like to come back to the questions that you had, the question you had in the beginning, which I think can be formalized between looking at the relationship between the p-values when you do a t-test and the loadings when you do your PCA. And so this figure shows exactly that. So this is the ALL data set. As we, one of the challenges asks to produce this plot. So the ALL data set with the 70 and something samples. The negative class is in red. BCR, ABL class is in black, along PC1 and PC2. And I think one of the questions asks whether the two first principal components are enough here to discriminate these two classes, and clearly here we don't have enough. So we would need probably to take more principal components to start to see separation between the two classes. And so we also did statistical tests to look for genes that separate these two. And so here on the right-hand side, we look at the correlation between the PCA loadings, so that would be for the first principal component, and our adjusted p-values. And we don't see any correlation. So this uses a smooth scatter. Most of our points are here, but we don't see any correlation between these two variables. So now let's repeat the same figure, but choosing only the features that have an adjusted p-values more than 0.05. So let's not look at all the features and all the loadings, but only those that have a small p-value. And they are actually 183 values. So after data, I extract the feature variable adjusted p-value, and I only select those that are smaller than 0.05, and I create this new data set for an ALL significant. I'll repeat my principal component analysis. So here they are. So if I limit myself to only subset of genes that are differentially expressed, I start to see better resolution between these two groups. But that shouldn't come as a surprise, because I do my principal component analysis using genes that show differential expression between the two groups. Okay, so I cheat, right? This is kind of self-put. And then indeed, now I start to see correlation between my adjusted p-values, and these are all small p-values, as you can see, from 0 to 0.25, and then my loadings. In my loadings correspond to here my first principal component that indeed starts to separate red and black group. Okay, red and black group. But this is not something that we can do in practice, because in practice, here we already know the results. In practice, this is what we have. We don't know which genes are differentially expressed. So we cannot use the loadings, so genes that are on the left or on the right, based on these, to try to come back and see which ones explain the difference between the two groups. So yes, if we know which genes are differentially expressed, and we use these to do the PCA plot, then indeed, PC1 here separates the two groups. So we have features that explain separation of these two groups. So yes, this is something that we can do, but not in practice, not when we don't know the results. When we don't know the results, the loadings explain the general variability of the data, and the loadings are independent of any groupings, of any annotation of the data. So in practice, we cannot use principal component analysis to try to identify features that are differentially expressed between groups. And if that's the question, to answer that question, we need to define the two groups and then look for differentially expressed features between these two groups. Doing my hypothesis testing is a supervised test. I know my black group and I know my red group. And I want to identify the genes that show systematic differences between red and black. In principal component analysis, I look at the overall variability of the data. What contributes to the overall variability of the data? Yes, surely there is differences in expression between red and black group, but there are many other variables that explain the global variability that I have in my data set, such as, well, there is noise in the data, there is gender, there is batch effects, when were the samples processed. So all these variables contribute to the principal component analysis. Are you satisfied with my answer?
So what I'm thinking about is, I came across this problem a few years back using a software package that a colleague had designed himself. And my recollection is that so exactly what I was asking you, so I agree with what you're saying, but I think, if I recall correctly, what we ended up doing was you take all the features and you remove one at a time, and you see at which point you lose the separation.
Okay.
And so that way you can start to identify the components that contribute most to the separation that you're talking about.
So that sounds something like feature selection, when you have very many features in your data set. Some explain the variability, some don't.
Yes.
So there are methods to do feature selection to keep those that are most informative.
Yeah.
Yes. Okay. That would be another type of question. And you can either start with all the features and then start to remove them and see you keep structure in the data, or you can start by adding features.
Yeah.
And as long as you add and you increase the variability in your data, you are happy.
Right.
And if you start to add things and they don't modify the structure in your data yet, then you
Right.
remove them. Yes. That would be a feature selection procedure.
Yeah, and I think that can be automated, right? Because you drew your bar plots of the PBE, right, the variations. And so there should be a way to be able to quantify that. And so you should be able to write an algorithm that maximizes.
Yes, so if you want to use principal component analysis to reduce the number of dimensions that you have, how many dimensions, how many PCs are you going to use? Well, one way would be to say, well, I use enough principal components so that I keep 90% of my overall variability as an input to another algorithm, for example.
Yeah.
Yes. And then your criterion would be, I want to keep 99% of variability or 95% of variability. So,
right. Or I want maximum of, say, 5 features. So what is the maximum variability that I can see for 5 features?
And by features, you mean your initial features in your data set?
Proteins. So let me put it this way. So I work in a clinical laboratory. I think about what new tests I can come up with for a particular purpose. And so from a practical perspective, on a particular platform that I want to use, say, I can only put down 5 different antibodies, or I can use 5 antibodies, and that I know, hypothetically, that's the maximum that I can do.
Yes.
And so that's what I'm...
But what do you want to, what's your question? What do you want to aim for with these 5 proteins, these 5 antibodies?
So I want to take the information from 5 different proteins to be able to, have a classifier that can separate or
separate what?
Or to identify a particular subset of patients.
So you want to do biomarker discovery?
Yeah.
Right? But then you would have to, disease and healthy. You would have, you would start with a shotgun experiment where you would quantify hundreds, thousands of proteins.
Sure.
And then you want to find which ones are those that discriminate the 2 classes.
Right.
Why would you not use a test that does exactly that?
No, I would, but are you asking why I wouldn't use
Why? So maybe we can carry on the discussion later, but my question is, why would you not use a statistical test where you have 1,000 proteins, 2 groups, and find the proteins that discriminate between the 2 groups?
Yeah, I can do that as a starting point. But
well, I suggest we carry on the discussion later, but why would you use something else? Because that is exactly what will answer your question. What are the 5 proteins that discriminate the 2 groups? But let's maybe...
and let me think further about
the
question.
Okay. And so the last part of plots that can be used to visualize high-dimensional data, or if we have kind of... are heat maps. So if we want to produce heat maps, there is a function in R called Heatmap to do that. So let me produce one heat map of the MOLVI data that we start to know now. Here it is. So here is the heat map of the MOLVI data. Without customizing anything, I take the quantitative matrix and I produce the heat map. And this is what we get from the heat map. We have 3 components. We have a clustering along the samples that we recognize. This is something that we did with the hclust function. We have our Zen group, and then another cluster with our 4872 points of the time points, and then another cluster that contains our early time points. There is also clustering that happens along the features. And here they are, we can't read them, there are too many of them. Okay. And these clustering will define how the columns and the rows are reordered. And then finally, the intensities here are replaced by colors. But if you remember what we saw earlier, is that and there are many parameters that can influence the result of clustering algorithm. So we looked at this on a relatively small scale. We had 3 genes and 5 samples. So let's see what is this effect on this kind of real data set heat map. So by default, in heat map, we use the Euclidean distance for our hierarchical clustering. And let's see how things look like if now I say use the correlation distance. And here it is. So we have different clustering along our columns, our samples. Right? We see here we have middle time points that are still together. And then we have here, early time points, but now the early time points, actually no, we have Zen and 24 hours that are form a cluster together. And then we have 16, 72, and then 0, 16, and then 72 and 48. I don't know if you remember the clustering along the features, but it does display a very different pattern here. Okay, it doesn't want to. Let me rerun it. Okay, right. Very different pattern in my feature cluster. So issues that we identified for the clustering, of course, have an impact on the heat map because we use the clustering. So the ordering of our columns and rows will be dependent on the clustering algorithm that we use, and of course on the distance matrices that we calculate. So there is actually another aspect that is crucial here in terms of producing heat maps, is how the colors are produced. So I'm not referring to the actual colors that we use, the color scale, and Stephen addressed this yesterday, but how are these colors produced? And if we look at the documentation of the heat map, we see that the colors are scaled. Okay. So let's look at our dummy example from before, where we had 5 samples and 3 genes. This is the heat map without any scaling. For this, E was the matrix, 3 by 5 matrix. So here's how it looks like. Now, when you produce your heat maps, you can scale it not the data as it is. So we are not going to change the data. We're always going to use this matrix here. But the scaling of the colors can be done along, for example, rows. This is what we get if we do the scaling of the colors along the rows. Here is what we obtain. Oh, sorry. This is the one without any scaling. The code is at the top. No scaling. So here the colors should reflect the original intensities that we have in the matrix. If now we scale along the rows, this is what we get. Look at gene 2. Look now, gene 2. Scaling along the columns. So these are, I mean, as far as I'm concerned, different heat maps. If I look, the point of a heat map is to look at it, and without having to think for too long, look at the image and get some kind of impression of the data set. As far as I'm concerned, if I look at this, or if I look at that, I have kind of very different impressions here and here. So I have to admit I'm not a big fan of heat maps. I don't think that heat maps are very informative. You use a lot of ink, lots of colors, kind of lots of eye candy, but one has to be very, very careful when kind of looking and interpreting these heat maps, because there are many parameters that will influence it. We have distance, we have the clustering algorithm, but that's true also for clustering. But in addition to that, we have how are we going to scale our data when it comes to colors, rows, columns, or no scaling. So yes, I'm not convinced about using, yes?
So by scaling, you'll be normalizing
Yes.
that particular row or that particular
Yes. So
there will be only one correct way to do it for a particular interpretation. If you're trying to compare across samples, then you would want to scale the particular row, right?
yes.
We can do both.
Yeah, there is no right or wrong, but depending on what
Message.
processing or, yes, what message you want, what processing you do, you will have different views. You will produce different figures. But if we move back here, what message are you trying to convey with a heat map like this? That's a question to you. So typically, I mean, I'm sure you're all familiar with heat maps. So what, and even though, I mean, you know more or less what this data is about. It's a time course, 6 time points, I don't know, 2,000-3,000 proteins. You have seen figures like that. So what is the information that the authors would try to convey with this heat map? I'm very critical of heat maps. So.
Basically, that a lot of variables you're changing across all the examples.
Okay, and what feature do you use to make that conclusion? There is no right.
I can't figure it out.
No, I think it's basically saying that Zen is very distinctly different than 0.8, 720,
Yeah.
16, and that it's very stark and looks nice in a figure. You can't really see what any of the genes are or the proteins, but that's the shock and awe of this kind of image.
But I find it useful to tell my collaborators, look, we should take this sample out of the data set, or this is a sample that's off. Can you go back to your notebook
Okay.
and figure out whether this replicate there's something odd about it. I dislike them, and I always thought it's because I don't understand them, and I don't know how to interpret them. But what I found useful lately is what you were just saying. When one thing just pops out as you, when everything else being the same, one thing pops out, you're able to go back and say, what is it about that row or that column that is so different?
About that column, yes, but about that
No,
row.
not row,
Yeah.
sorry. Column,
Okay,
column.
but so I agree with you, but do you need a heat map to make that conclusion? Would it not be,
I'd love to have a better way to show it.
so would it not be useful to only have the sample clustering? Because the sample clustering tells you that these 3 samples are very different
Yeah,
from the
but
rest.
collaborators, I don't think very often understand tendrils very well. And
Or PCA analysis.
Oh, I'm more scared of PCA than of heat maps. I'll just put it out there.
However, it's definitely done within PCA.
Okay. Times. So something like this, a PCA plot, 50%, 21%, showing that kind of time point 6 here, 1, 2, 3, 4, 5. To me, this tells me exactly what I want to know without all the kind of bright colors of the heat map.
Would you be, go ahead.
Oh, I was just going to say that with the heat map, you're also getting an idea of what features are sort of different for those samples, right?
So, which ones are the features that?
So like for the first branch or for the first group of samples, so you can see that.
These features?
Yeah.
So which ones are these features? Do you know?
I mean, if it's a better heat map, you can tell them.
No, when you have thousands, you will never be able to read them. We'll see later.
Yeah.
But if that's your question, then you could do a statistical test to see which ones are the features that discriminate this group from time point 0, things like that. Anyway, it's not for me to say. What I want to say is that I agree with all the observations that you made, saying that this heat map tells us that Zen is very different from the other ones. But I think that the information that we actually use to make this is what we see there and there.
Can I?
Yes.
What if one within Zen was very different?
Okay. Yeah.
So you had one row that was really out. In the PCA plot that you showed, would that be like one of those black dots would be
Yes.
in the middle of the graph, and that would be your way of saying, look, that particular column is off.
Yes.
I just want to remove it so that I have 2 dots up in the, that would show up in
Yes.
the PCA plot.
So here we see that the time points always cluster together. And actually here we have it along 2 dimensions rather than in your hierarchical clustering is along 1 dimension, it's the distance, the height of the tree. And so yes, we would have one of these replicates that would be away from the 2 other ones.
And would you be able, from this graph, to say which of the 3 replicates is the one that's off? Because that's one
Yes.
thing that I find useful when you have the heat map, is you can say it's time point 2 or sample A of time point 2.
So I will explain this code a little bit more later on, but let me say
Perfect.
I'm going to update this plot a little bit. And then text. So here I add the numbers that are stored in my object, and so replicate 1, 2, 3. Here we don't see it because it's black on black.
But you could figure out which one it is.
Yes, you could.
So I need to learn more about
You-
PCA plots.
You could. So, yeah, I think this is a bit of an information overload without being able to extract easily information. Yes?
But I think we should add that PCAs are informative if they're explaining a major amount of variance. Like sometimes you see PCAs which are saying like 2%, one of the PCAs is 2%, one of the
Yes.
PCAs is 1%. Then it doesn't really say anything because you're reducing all that data to them.
You are absolutely right. And in this case, we see that this PCA
Yeah, this
over 70% of variability. So this is meaningful. If indeed we had 2%, if we had, these are 18 dimensions. So we do really well 70%. We do really well 70% of 18 dimensions. if we had 200 dimensions, it's very unlikely that the two first principal components would explain that much. But then we need to be very careful how we do the interpretation. So it's important to look at the percentage of variance explained for the two PCs. And most likely, if we didn't explain that much variability, we would not have such clear separation. Sorry, I think there was a comment or question at the back. Yes?
I guess just a question about the stage set. So it's saying like replicate 1, 2, 3, Z. So these are biological
Yes.
replicates?
Yes.
So I guess in response to Kurt's question, we need to make sure that like, you should have technical replicates before you throw any data out to begin with. Because like if you're saying that one of your Zen biological replicates was not clustering well, like maybe there's something important there, and so you shouldn't just throw it out.
So if well, if you can run technical replicates and biological replicates, why not? But the most important ones would definitely be biological replicates. But let's say that I have much more variability within each time point, more biological variability, so that, for example, we see structure within time point 1, 2, 3. But imagine that this would be kind of the red, blue, and greens one would be would not form these clusters. That would not be a problem at all. It means that the biological variability within time point 1, 2, 3 is as big as the variability that we observe over time. That is not a problem if that is the biology. And then same thing, maybe imagine the biological variability is bigger so that orange and yellow start to overlap. Again, that's fine. But now let's imagine that we have this structure, but that one of these suddenly is here. That would be weird, because don't forget that PC1 explains 52% of my variability. PC2, only 20%. Now, okay, so now suddenly if one of these points were to move here, that would be a huge change. So suddenly one of the Zen replicates would be closer to the 72 hours time point. Now they are still separated along the second dimension, but we see that PC1 retraces the time course to some extent. So that would still be a huge variability for this cluster compared to the other ones. Now maybe there are reasons that this last time point is more variable than the other ones, and that will depend on the biology, on your experiment. But maybe one assumption is that within each group I have similar biological variability. If not, this will have consequences if you start to do statistical tests down the line, because your variability in one group is very different than the variability in another group. Again, there is no right or wrong if that is what the data shows, but exploring your data, visualizing your data, will tell you whether something is maybe wrong or not. And something that we didn't address specifically, but it's always good to also look for batch effects. So here we have nice matching between the experimental design and the clustering, but this is not necessarily the case. And it's always good to also track things like who generated the data when you have different operators, when the data was generated, because these batch effects could explain most of the variability you have. Yes? Yes, yes. And so I cheated a little bit on one of the things you said. Here, when you said we can see features that are differentially expressed, and so I was a bit naughty. I said, oh, which features? These ones, which ones are these? Well, actually, as it happens, there are packages, for example, Heatmaply, and D3 heat map. And I'll leave you try this out. So if you run this code exactly, these will allow you to produce interactive heat maps where you can go and select the rows of interest. So it would be possible to select these and look at the gene. So I was a little bit unfair here. But before you do that, I also want to mention other packages that allow you to produce heat maps. And here's one example. And you can either try the interactive heat maps or this package, which is complex heat map from Bioconductor. And so what I do here is that I create annotations. So for example, using the multi-data, I do some hierarchical clustering, as we did beforehand, and extract. Here I choose 12 clusters. So I cut my hierarchical clustering to have 12 clusters. So this is one thing I use to annotate my rows. Okay. I'll also use the time points to annotate my columns. And I also use here another annotation where I produce a box plot of intensities along my columns. So this is another annotation. And then I create my heat map with my top annotation, bottom annotation, and side annotations to produce this. Okay, so here I have my time points, Zen, and so on. And so here I see that my annotations match exactly the clustering that I have. And here I have my annotation of clusters. I happen to have one really big cluster. Okay, and here's my kind of box plot notation. So heat maps are very powerful, of course, and they can be used to do lots of annotation, as shown here.
What did the bottom box plots show? The distribution of the data with regards to like the distribution of the ratios, or the expressions of?
It shows exactly here. Box plot. Of each sample, so my intensities in my different columns. Okay, but they are reordered here based on the clustering. So before we move on the visualization of mass spectrometry data with kind of dedicated packages, I'll give you a couple of minutes to explore the heat maps. This is one of my favorite packages to produce complex heat maps. This would be complex heat map from Bioconductor, or quite nice to try Heatmaply or D3 heat map. And this code is not executed, but I tried it. So if you run exactly this, you install D3 heat map and then run it like this, you will get a new tab that opens, and you will be able to kind of explore, zoom in and out from your heat map to, for example, find the features that are of interest. So now we're going to focus on a set of packages that come from the Bioconductor project. And for those that were here last week and that took the advanced R course, where you learn about package development and object-oriented programming, these are exactly the kind of skills that are needed to produce these packages that eventually could be submitted to Bioconductor. And so currently we have 1,560 packages in Bioconductor. Most of these are probably targeted towards kind of genomics. But if you follow this link here, you are then redirected to a page that lists all of the packages here, 1,500 plus packages. But here on the left-hand side, you have a set of keywords, or also called BioC views, that allow you to specifically look for subsets of packages. It's a little bit like the filter in dplyr where you say filter equals proteomics. Well, here you could use this interface to say proteomics, and it will then subset packages that are related to proteomics, that have some sort of relation to proteomics data analysis. And if we look at, okay, so 105 here, 105 packages, 105 of the 1,500 packages have this tag, this annotation proteomics. Similarly, another one that is of interest would be mass spectrometry. Okay, there are here 70 entries, 70 packages that are annotated as being useful in the frame of mass spectrometry. Okay, so there are many packages out there, but they are annotated, so there is a kind of relatively, or it's easier entry point in identifying the packages that might be useful for you. So I will be focusing on one package in particular here that allows you to read raw data, or different types of mass spectrometry data in, and kind of manipulate and visualize them. This is the MSN-based package. To demonstrate this, I'll be using data that is distributed with a package called MS Data. And to see what raw data sets are available, we can use this function here. And this function prints out a set of data sets, raw data files that are available. We have MRM mix, we have an MS3TNT10 data set, an MS3TNT11 flex data set, and some other TNT data sets. All in MZML, yes, all in MZML format. And with this function here, we can get identification results in proteomics in MZ-identML format. Okay, so these are some of the data sets that I will be using to kind of illustrate what can be done. And then another one here, data, iTrack data. So this is a small data set that comes with MSN base. These other ones are distributed to MS Data. And this is a kind of pre-formatted raw data object. So we have essentially in mass spectrometry-based proteomics 3 types of data sets. We have raw data that generally comes in the form of MZML or MZXML, NetCDF, MZData are also supported. And we have a second type of data set which are identification results, and these generally come under the format MZ-identML. And finally, we have quantitative data that come as spreadsheets, for example, CSV or MZ-tab format. Are you familiar with the MZ-tab format? Anyone? So I won't spend too much time on this format. It's a kind of tab-delimited format for quantitative proteomics or metabolomics data. But it's somehow surprisingly not yet well established. So raw data, identification data, and quantitative data. And so once we have raw data, we can read these in with a function called read MS data. This is the function in R that will allow us to read raw data and produce something called MSNX. So it's a specialized data structure that is fine-tuned to read raw data and allow us to parse or kind of manipulate raw data. There is a function called read MZ-ident. Oh, no, sorry, I think ident data.
Let me check. Read MZID data.
That takes MZID file as input, and this file produces a data frame. Oh, sorry, this function returns the data frame. That parsed and processed the data here. And so if we have a CSV file, for example, one that is exported by Max Quant, Proteome Discoverer, and the like, there is a function called read MSN-set2, and that one will produce an MSN set, like the MALV 2015 data, like the Hyperlipid data, and so on. So these are the three functions that will allow you to read different types of data in. So in the notes, I show detailed examples of how to read data in. But if you're interested, all this code can be re-executed. But I will move immediately to the visualization aspects. So there are sections on how to read in raw data, quantitative identification data, how to integrate into identification and then the raw data, and how to read it quantitation data. And once we have raw data, we can extract the chromatogram, the total ion chromatogram, or the base peak chromatograms, and produce these figures. To do this, let me show you. Okay. So if RW is a raw data object that was ready with read MS data, extracting the chromatogram here to create this CHR chromatogram function will allow us to plot the extracted, also the chromatogram. And this would also work if we had multiple files. So these examples only read a single raw data file, but you could provide it a string or a character of tens or so raw data files. And this would work on the laptop, or even tens or hundreds of data files. So here I show the following example where I read this MS3 TMT11 dataset. Okay, so here we have three levels of mass spectrometry: MS1, MS2 for the identification, and then MS3 for quantitation. That is a TMT11 plex experiment. So this is the file of interest, and here I read this raw data file. This is the name of the file.
Why are you using just like MZML rather than the MZXML?
So MZML is a more recent format,
Okay.
and it's kept up to date. I don't think MZXML is capped with the new CV terms. So I would, in general, use MZML, but some of my older files are in MZXML, and as long as these functions are concerned, it doesn't matter. You would expect exactly the same behavior. And same thing if you used MZ data on NetCDF.
Are there more features for one versus the other? It doesn't really
At this stage, it doesn't matter, but of course, if you have specific annotations that are only present in MZML, for example, then if you export your data into one of the formats that don't support these CV terms, then they will not be available. But packages are written so that you would use the same functions, the same syntax, irrespective of the file, the original raw data file. So here's this raw data file, and it tells us that we have this 990. So that's a sliced part of our raw data file. We have just under 1,000 spectra. We have like MS levels 1, 2, and 3. And here, so we can ask for specific properties of this data set. So for example, if I ask which ones of these spectra are centroided and which MS level do these spectra correspond, I produce this table that tells me that MS1 spectra, 30 MS1 spectra, they are not centroided, they're in profile mode, whereas my MS2 and my MS3 spectra are centroided. Okay, this is the acquisition numbers of my spectra. They have this kind of F1.S001 a naming convention. F1 corresponds to the file, so here we have only one file, but we could have read multiple files. And so that's the first spectrum on the first file. And so we can start to manipulate this raw data, for example, by asking, well, let's filter the precursor scan 21945, which is this one. And what the software is going to do is going to say, okay, well, this spectrum here is actually an MS1 spectrum. So it's going to automatically identify which are the MS2 spectra that stems from this MS1 acquisition, and which are all the MS3 spectra that stem from these MS2 spectra. So it create a kind of sub-experiment with all the kind of offspring spectra from this one, because that's an MS1 spectrum. We could also have used here an MS2 spectrum, the acquisition number over the MS2 spectrum, and then it would have looked for all MS3 spectra as well as the parent ion, the MS1 spectrum. So here we create a kind of a subset experiment with 35 spectra. And then we can start to extract spectra of interest. So if we extract the first spectrum, in this case it was a quiet attention time 45.7. It's an MS1 spectrum, and we can plot it. Double square bracket operator for subsetting raw data to extract a spectrum. That's the same syntax as you would have for lists, for example, in R. And here it is. So this is the spectrum object, and then if we plot the spectrum object, this is what we get. Retention time in seconds, and here's the spectrum. This is how it's plotted. We could, of course, extract the individual MOZ values, the individual intensities. If here we extract the third spectrum, it's not an MS1 spectrum, so it's part of the kind of spectrum 2 class. Precursor 599, which was the one before. This retention time is MS2 and additional features. And if we want to plot it, here it is. Plot RW23, the third spectrum, and here it is. So this is the precursor MOZ. So this is the MS2 spectrum that originated from the first plot that we saw. And then here, this is an MS level 3 spectrum. And more specifically, here we focus on the TMT11 range. So they overlap here. But these are the reporter ions that are used for quantitation. And these are the reporter ions corresponding to this so we can do additional visualizations. For example, here if we extract a set of MOZ values of interest, a set of retention times of interest, we can produce this MS map. And the colors, default colors are excellent, but I'll present it here as intensities in 2 dimensions. If you don't like the 2 dimensions, but you prefer the 3-dimensional plot, here it is. So same M over Z range and retention time, and here we see the features. Similar plot, but where we zoom in between 2 MS1 spectra, and this is the set of MS2 spectra from that channel. This MS1 spectrum, and here are the 10 MS2 spectra were acquired by some of these precursor lines. And if we repeat this multiple times and visualize these 3D figures in a row, we can then produce these animations where here we have a constant M over Z range, and then we have different slices of retention time, and this represents the way the data was acquired. We can also, so here's another example of raw data. It's this file, MZML. So I read this file in. I specify that it's not centroided. This can sometimes be automatically detected from the raw data file, but not always. And then I define a specific M over Z range and retention time range that is of interest. So here I will be specifically look at the serine, and here's the serine M over Z value. And then I'll identify an M over Z range, a range between the serine M over Z values and the retention time range. Okay. So my raw data is in profile mode QAs. So I can use the piping to filter along retention time and filter along M over Z range. There with these 2 functions, and then I carry on piping like we did with deep learning. So this generates a kind of new raw data object, but it is specifically focused around the serine region.
Are these types of data tidy always, or do you have to manipulate it? Is the input you put to these are, I'm sorry, MZ or MXL?
So these are not data frames. These are not data.
Able to use dplyr.
Yes. So the original concept of tidy data was developed around data frames. And the idea of tidy data and data analysis is that I have a data set where I know where to find what. And in terms of a data frame, this means observations along the rows and variables along the columns. And then if I use tidy tools with the pipe, tidy tools are tools that get a tidy data set as input, and then they will return a tidy data set as output. And that assumption allows us to pipe through different filtering functions. Now, we don't have data frames here because this data doesn't really fit in a data frame. We have another type of data structure that is shown here. And this data structure is called an MSN experiment data. It doesn't fit in a data frame. It contains all sort of slots, feature data, phenotypic data, as we have seen before. And it manages all this raw data for you. So it is, to some extent, a tidy data, because we know where to find what as expected. Irrespective of the file that we use, this data structure will handle the data complexity for you. And so if we move back to the filtering here, these are tidy tools, because these functions take this specific tidy data as input and then guarantee that they will return the same data structure. And so we can chain them as we did with deep learning. So the principle is the same, but the data is not a data frame anymore. These are these other special functions. And they behave the same way. What is fed in by the pipe is used as first argument. And so we can plot this experiment as an extracted ion chromatogram. Here it is. So here is our retention time dimension. Here's our M over Z dimension. So here's the extracted ion chromatogram. Okay, and each point here represents a feature, a data point. And we have this bump here around the serine, expected M over Z value. And here is one specific retention time. So it's kind of a three-dimensional feature. I can do my extracted ion chromatogram, specifically at this retention time here. Extract my spectrum at that specific retention time, and this is output slot, next one. So this is in profile mode. We can then use specific tools like pit peaks to start with my profile data and produce centroid data, repeat my filtering in retention time M over Z, repeat my XIC plot, and this is what we get after. So it's not perfect yet. We do not have all our peaks that were picked, that were chosen at the expected M over Z value, but these tools allow us to kind of optimize our peak picking algorithms. And there is a link here to one of the vignettes that describes how different algorithms can be chosen to optimize the peak. And so that was some aspects of visualization on raw data. We can manipulate spectrum data or peptide sequences. So for example, we can calculate fragments from a peptide sequence as is here. And so calculate fragments of this peptide will calculate all possible A, B, C, X, Y, and Z ions that can be observed for this peptide. Now we happen to have a spectrum that we know matches this peptide, and so we can plot, so it's the I spectrum in this raw data set. So we can plot it and also provide the sequence, and software here will annotate the spectrum. This happens to be an all-plex, so here these are the 4 reporter ions, but the software will annotate the spectrum automatically. If we want to plot two spectra, 41 and 41 spectrum, the 42nd spectrum is archive data. These happen to be 2 spectra for the same peptide. We can produce these kind of visualizations where the matching peaks between 41 and 42 are high. Briefly, there is a MSGF GUI package that allows you to do additional visualization and exploring these identification results. I think in terms of visualization, we shouldn't miss out MS imaging. So there are 2 packages I'm aware of for this. There is Cardinal, and if you have questions, I suggest you ask them to Kylie. So here's the slice of what show this is.
Fetal pig.
There we go. And here the reconstructed image for this M over Z value for this feature. So bioconductor packages, BioC-Lite, Cardinal, and there is a link to the web page. And the second package I'm aware of is MultiQuant, very similar. 3 images corresponding to 1, 2, 3 M over Z values. These 3 features. Missing values. So there is an image2 function that is like the image function in base R. The only thing that it does is that it does a better job at labeling samples and numbering features as opposed to image. So here we have missing values in white and intensities in yellow to red. Not very useful, but we can pass this is.na that asks, are any values in this MSN set missing? And so here we get a quick overview of values that have cells that have an intensity in black and cells that have missing values in white. And a plot that I find very useful. As before, we have an image with our samples and our features. But what this function also does, it counts how many missing values I have for all my rows and how many missing values I have for all my columns. And then it's going to reorder these rows, columns, and rows to display those at the very top that have more missing values. So here's a bar chart up to 11 or 12 missing values, and then we have some 10 missing values, 9, and so on. And same thing, if we reorder the columns, these are the samples based on how many missing values we have. So it doesn't identify which features, but that would be relatively easy to calculate. But it tells me the proportion of features that have a high number of missing values and gives me a distribution of missing values among my samples. Maybe there is a sample that has a very high level of missingness. So I wanted to finish on the PCA plots because using Amazon sets, because PCA plots are things that I end up doing a lot for the kind of research that we do in my group. So plot2d is a function from the Prolog package that will automatically know what to do to produce PCA plots either from proteins or from samples. So this is the multi-data, this is the hyperlogic data. Something that I haven't mentioned, but if you have missing values, the PCA analysis will not work if you have missing values. So if we use plot2d, here it's going to automatically remove proteins that have missing values. Okay, so it tells you that it automatically removes 388 proteins that have one or more missing values. Now, if we use Amazon sets, we can also do something like that manually with the filter NA function. So these two will have the same result in terms of visualization. Here, the missing values are removed automatically. Maybe I wasn't aware that I had missing values, and here I do it manually. Here I could even parameterize what features to remove based on the proportion of missing values that I have. And actually, using the plot2d provides different types of dimensionality reduction techniques as a method here. So here the default is PCA, but here I could also put T-SNE or NIPALS, which is an PCA implementation that will support missing values. So with the same function, with or without filtering and by specifying the method, it becomes relatively easy to produce many PCA plots, and it will automatically label them. The percentage of variance explained by different principal components, and by default it prints PC1 and PC2, but this could also be parameterized. So are there any questions about mass spectrometry visualization, what we have seen here? So we now have a refreshment break from 3 until 3:30. And so the last session is free practice. So there were a bunch of exercise challenges. Some were relatively easy and others were a little bit more involved. So you're welcome to come back at 3:30 and either work on your own on some of the challenges or ask me or us, and we can either help you do the challenges or I can do some of the answers of the challenges here. Okay, or we can revisit some of the material if you still have questions. Okay.